Okay, can you see my slide?
Yes, perfect.
Okay.
Thank you.
Thank you for kind introduction, and I'd like to thank the organizers for giving me this opportunity. Okay, I'm our first speaker, so I'd like to introduce this session briefly. So this is an evolution before and after Luca. Doesn't work. Okay, I'd like to show you a figure from a famous review by Gerald Joyce. This figure shows that the evolutionary scenario of emergence and evolution of life from a foundation of Earth to prebiotic chemistry and our new world and present day DNA protein life. So most speakers in previous session mainly focus on around here, prebiotic chemistry to our new world. But speakers in this session mainly focus a latter part of the scenario around here, RNA world to present day protein DNA life. So this period is also very important, I think, because during this period, chemistry produces many biological phenomena. So we'd like to understand what happened in this period, but how can we understand what happened in this period? I think that speakers in this session try to answer this question by different approaches, such as mathematical model and simulations by Philippe Gay and Wendell Ma, and or using experimental model by me and Ryo Mizuchi, or phylogenetic analysis of extant organisms by Fei Ping Wang. So in the next few slides, I'll show you their studies briefly. The first speaker, Fei Ping Wang, is trying to analyze the archaea metabolism, mainly focused on alkane metabolism. And recently, she shows that the first archaea has very simple alkane metabolism pathway, and methyl reducing pathway. And then it's developed to a more complicated CO2 fixation pathway. And then they develop modern beta oxidation pathways. I think that such a phylogenetic analysis is a very powerful tool to understand the evolutionary process after Luca. And the next speaker, Philippe Gay, recently published a very important article, which shows that all catalytic reaction can be categorized into just 5 types. That's very surprising. And also in his recent paper, he presented an idea of multi-compartment autocatalytic reactions shown here. In this idea, the one compartment contains only an incomplete autocatalysis, but when it's combined with another compartment with a semi-permeable membrane, then the reaction system as a whole became autocatalytic. I think that such a much compartment autocatalytic reaction can enhance the possibility of appearance of autocatalysis in prebiotic Earth. The next speaker, Ryo Mizuchi, he's a member of my laboratory. Will present a recent work over RNA coevolutionary system, and he found replicating RNA spontaneously develops the complexity, producing the replication networks. And I'll mention about his finding in my talk later, just briefly. And the last speaker, Wendell Ma, again, analyzing the possible evolutionary process using computer simulation and mathematical modeling. And he gave me a long list of his recent works. I read some and found that I show you two interesting results we I obtained from his works, membrane permeability of raw material is critically important for development of protocell, and also chirality may be established at polymer level before the advent of polymerase. That is very surprising for me, and I'm not sure he will talk about these studies, but if you have interest, please read these papers. So now let's move on to my own talk. Today, I'd like to talk about host-parasite coevolution of RNA protein replication system. So here, this picture again, and our interest is Darwinian evolution. And I think that Darwinian evolution started around here, probably before RNA world. And then the Darwinian evolution produces many biological phenomena, I think. But I'm wondering, is Darwinian evolution sufficient for emergence of life? I think that it's necessary. Darwinian evolution is necessary for the emergence of life because without evolution, there is no way to produce complex living things, but I'm not sure it's sufficient. I think so because we know the other replication systems that are capable of Darwinian evolution, but does not produce living things. The first example is the Spiegelman's RNA replication system reported in 1967. In this system, the Spiegelman's group uses RNA genome of bacteriophage Q beta and also use the RNA replicase, also that originated from the same bacteriophage, and they performed 74 transfers and found the RNA eliminate 83% of its RNA sequence, so it became shorter and replicate faster. Another example is Wright and Joyce's work using the ribozyme and reverse transcriptase and RNA polymerase, and they performed a 100th transfer and found that 29 mutations accumulated into the ribozyme, and the ribozyme became replicate faster. So in both cases, RNAs became replicate faster. It evolved, but no biological characteristics appeared, such as diversity and complexity and new function didn't appear. So our question is, how does Darwinian evolution produce biological characteristics, such as diversity, complexity, and cooperation, and so on? So something is lacking in the previous RNA replication system. So what is lacking? I think one of the lacking possible answer is translation. This is based on the Luis Milazzo's studies. It says phenotype-genotype decoupling, that is exactly translation, is a key to open-ended evolution. So according to this study, we first tried to introduce translation machinery into Spiegelman's RNA replication system. So we constructed a translation-coupled RNA replication. In this system, RNA replicates, and we called it the artificial genomic RNA. It's about 2,000 nucleotides, and it's encoded only one gene, RNA replicase gene. But more precisely speaking, it's a catalytic subunit of the replicase. Now, this is the same replicase used in Spiegelman's case. And another factor, it's different from Spiegelman's system, is a cell-free translation system of E. coli. This is a reconstituted translation system. People call it a pure system. It consists of the protein and RNAs that is required for translation, such as ribosome, tRNA, amino acid, NTP, and so on, all the purified. So the largest difference from Spiegelman's reaction is this cell-free translation system. In the Spiegelman's case, RNA replicase is supplied as a purified protein, but in this case, the RNA replicase is translated from the RNA. So translated replicase replicates the RNA, and mutations are introduced during this replication process. So to make this system replicate continuously, we need another factor, compartment. Compartment is required because to make a linkage between RNA and the translated replicase, and also to repress the excess amplification of parasitic RNA, I mentioned later. As a compartment, we use the water in immersion. This is a very tiny droplet, bacterial-sized droplet. You can easily prepare just mixing reaction mixture in the oil and the detergent. So inside the droplet, we incubated the droplet at 37 degrees for 5 hours, and inside the droplet, translation and replication occurs, and the aliquot was diluted with new droplet containing new cell-free translation system to continue RNA replication. This is one round, and we repeated this serial dilution cycle for many generations. Now, what happened after a long-term serial dilution experiment? Today, I show you 3 results. First, parasitic RNA appeared and co-evolved with a host RNA. And second, host RNA was diversified, probably due to the co-evolution. And third, evolved host RNA tend to incorporate deoxyribonucleotides. So not ribonucleotides, deoxyribonucleotides. First result is parasite. So here I show you a trajectory of RNA concentrations of a long-term serial dilution cycle. You see the genomic RNA concentration is shown in blue line, and you see that at the first stage, parasitic RNA, the red marker and the red line, appeared soon. It's a very small RNA, about 200 nucleotides, and it's produced from genomic RNA by deleting internal replicase gene, probably through RNA recombination. And this small RNA does not contain replicase gene, so it cannot replicate by itself, but it can replicate if it is in the same compartment with other genomic RNAs using the replicase translated from genomic RNA. In this sense, this is a parasite and this is a host. So once parasitic RNA appears, the RNA concentration decreased because of the parasite. And after sufficient dilution of the parasitic RNA, the host RNA again recovered. In this way, oscillation pattern appears. And in later stage, the oscillation pattern gradually changed, became unclear, probably due to the parasite resistance was acquired by host RNAs. In this way, replication proceeded, and in the final stage, we found that 2 types of new parasitic RNA appears. The green one is a long parasite, about 1,000 nucleotides, and the purple one is a middle-sized parasite, about 500 nucleotides. These are new parasites. We isolated these RNAs, but they don't replicate by itself, but it's replicated only combined with host RNA. So now we found that various types of parasitic RNA appears. And parasitic RNA is not RNA, parasites is a well-known problem for replicators. Here I show you some literatures. Theoretically, parasite has been considered as a serious problem in self-replication systems. I think that the first example is Manfred Eigen's hypercycle model. This is a parasite. Once parasite appears, it destroys hypercyclic replication. And in later studies, many theoretical biologists include the parasite in their systems. And it is also known that a parasite may be inevitable for all life and replicators. And actually, in some experimental model, parasitic behaviors are found. And also, it is known that parasites affect host evolution, inducing evolutionary arms race, diversification, etc. So Based on these literatures, we saw that coevolution with parasite may induce diversification of the host species, host RNA species. So to investigate the diversity, we performed a deep sequence analysis. First, extract RNAs at each point, and then perform the long-read INGS, next generation sequencer using PacBioRS. And we collected top 3 major genotypes at each point and draw a phylogenetic tree. So this is ancestor, and color of the dot represents the stage of the evolutionary experiment. The light color means the early stage, and the deep color means the later stage. And you see that there are two large branches, and the evolution proceeds into 2 ways. So this shows that host diversified into 2 lineages, and probably due to the coevolution with parasite. And I think that this is caused by the different resistance of each branches to different parasite. To test this, we performed the competition experiment using the one of the RNA on this branch and using one of the RNA on this branch, and mixed with 2 types of parasites that appears in later stage, green large one and purple middle one. And then we found that the RNA on this branch is relatively resistant to purple parasites, but RNA on this branch is relatively resistant to green parasites. So they have a different parasite resistance. Probably it is one of the reasons for different diversification into different lineages. Then now we continue the coevolution experiments for more generations. And in the previous slide, I show you the data before this point, 120 rounds, and the Ryo Mizuchi performed another 120 rounds of replication. This will be presented in the later talk, and he found that the diversified host RNA start to form a replication network. And also now we continue furthermore, and here I'll show you unpublished data. This data is just obtained today, so evolution continues, and you see that the oscillation amplitude gradually changing, gradually decreasing. In the later parts, they're very unclear oscillation. Please keep in mind, I mentioned this later. So in last few slides, I'll show you another evolutionary experiment. In previous experiment, serial dilution experiment was performed by manually. We prepared the droplet every day and incubated and diluted every day. Oh, wow, every day. So it's very laborious. So so we developed an automated droplet reactor. It is a kind of flow reactor, but the droplet flow reactor. So using this system, we performed a very long-term evolutionary experiment independently from the previous manual experiment. And you see that the very sharp peak here again, host RNA is shown in blue line, and we found that parasite always co-replicated with host species. So here I show you the number of mutations accumulated in the host RNA. It's increasing roughly constantly. So evolution continues. But this evolution is significantly different from previous manual evolutionary experiment in the phylogenetic tree. In this case, the phylogenetic tree is very straightforward, only one-way evolution, unidirectional. There is no large branches. So that is one different point. Another different point is frequent extinction. In previous manual evolutionary experiment, we have never experienced extinction. it was very robust. We continued the serial dilution experiment for more than a few years, but never extinct. But in this case, extinction frequently occurred in later part of the evolution experiment. For example, the extinction point is shown with red triangles. For example, at this point, we for the first time experienced extinction. The RNA concentration decreased under the detection limit and never recovered after a few rounds of RNA replication. So we went back a little and restarted RNA replication, then succeeded in the RNA replication. In this way, when we experienced the extinction, then we went back a little and restart. In this way, we continued RNA replication. But at the final point, we tried several times, but never succeeded in continuous replication. So we gave up. In this case, I'm not sure why extinction frequently occurs in this case, but the key, maybe the hint is this amplitude of oscillation. In previous manual experiment, the amplitude of oscillation gradually decreasing. It became stabilized. But in this case, it's very large. Maybe it's increasing amplitude. It's more than 4 order of magnitude. So it may cause a significant decrease of RNA population that may cause extinction, I think, but I'm not sure presently. But I think that the difference from the automated system and manual system may provide a useful insight to understand the stability of the replication system. Okay, here is the last result. It may be interesting. So I think that one of the next important challenge for RNA replication system is a transition from RNA to DNA. So for the transitions, the RNA polymerase we are using have to synthesize DNA. Then we examine we compare the DNA synthesizing ability or deoxyribonucleotide incorporation ability of the RNA polymerase that appears in the course of evolution. The first RNA polymerase, that is a wild type RNA polymerase, does not incorporate DNTP because it is RNA polymerase. There is not DNA polymerase, so it's reasonable. But in the RNA polymerase that appears in the course of evolution gradually start to incorporate DNTP. But the ratio is very low, so therefore it's not a DNA polymerase, it's still RNA polymerase, but the incorporation rate increasing. There is a clear trend. So this is a little surprising for me because there is no advantage for RNA polymerase to incorporate DNTP in our system, because in our system, we didn't add DNTP. Our system does not include DNTP. But I think that the possible explanation for this result is replicase might relax substrate specificity because it is unnecessary. I think that is original RNA polymerase usually works in the cell, and in the cell there is a large amount of DNTP. So the wild type RNA polymerase have to distinguish between NTP and DNTP. It's essential. But in vitro conditions, in vitro evolutionary conditions we used does not contain DNTP. So in that environment, RNA polymerase does not need to distinguish DNTP and NTPs. So it's lost their specificity because it's unnecessary. That is our idea. So then RNA polymerase start to incorporate DNTP. Irrespective of the reasons, I think that this trend may imply that the transition from RNA to DNA may be easier than we previously thought. Okay, here is a summary of my talk. Manual long-term serial dilation experiment induced parasitic RNA appearance and diversification, and automated long-term serial dilation did not induce diversification and cause frequent extinction. I'd like to know the critical parameter. And RNA polymers that have evolved in our in vitro system start to incorporate DNTPs. Okay, the result I presented today is a collaboration with a member of my laboratory and also previous Professor Yomo's laboratory members, and also supported by these funding. And I'd like to thank them all. And also I'd like to thank you for your kind attention.